Good morning all. Today we are going to discuss about the nutritional classification of bacteria. The nutrition is a substances used in biosynthesis and energy production and therefore are required for all living things. These points we have discussed yesterday. Bacteria, like all the living cells, they require energy and nutrients to build up their proteins and structural membranes and also to drive their biochemical process. So for all this, they require energy and nutrients. Bacteria require source of carbon, nitrogen, phosphorus, iron and a large number of other molecules. The carbon, nitrogen and water are used in higher quantities. And the nutritional requirements for the bacteria, this can be grouped according to the carbon and energy source. Some types of bacteria must consume preformed organic molecules to obtain their energy, while others can generate their own energy from inorganic sources. So these are the two types of bacteria which consume preformed organic matter, or other bacteria can generate their own energy from inorganic matter. So this is the classification in a brief. All organisms, they are classified based on the energy source into, that is in the form of the energy source chemical or the energy source light. So this, according to this, it can be divided into chemotrophs, and if it is under the light is used as an energy source, it is called phototrophs. Again, these chemotrophs are classified based on the carbon source. For example, those are using organic compounds are called chemoheterotrophs. Those are using carbon dioxide are called chemoautotrophs. And as a result of this metabolic process taking place in chemoheterotrophs, and this takes place with the help of the final electron acceptor, and they may release oxygen. All the animals, most fungi, protozoa and bacteria are what? They are chemoheterotrophs. Then in the coming under the chemoautotrophs, hydrogen, sulfur, iron, nitrogen and carbon monoxide oxidizing bacteria are required by them. Again, the final electron acceptor, if it is not oxygen, if it is oxygen, then it can be used by all animals, most fungi, protozoa, bacteria. If it is not oxygen, this can be divided into organic compounds and inorganic compounds. Organic compounds are fermentative, for example, streptococcus. Inorganic compounds are electron transport chain, that is Clostridium, for example. And coming to this energy source as light, can be divided into phototrophs. Based on the carbon source, they are again divided into organic compounds and photo, that is carbon dioxide. And again, based on this, utilizing this carbon dioxide, the photoautotrophs can be divided into those who use water to reduce carbon dioxide. So the photoautotrophs are again classified again according to the organic matrix, organic compounds. If they are utilizing organic compounds, then it may result in photoheterotrophs, that is example green non-sulfur bacteria, purple non-sulfur bacteria. And in the case of the photoautotrophs, if they use of water to reduce oxygen, they can be divided into photooxygenic photosynthesis. Those which reduce oxygen are called anoxygenic photosynthetic bacteria, example green and purple bacteria. So these are the brief classification. On the basis of this energy source, the organisms are designated as phototrophs and chemotrophs. The organisms which can utilize light as an energy source are called what? Phototrophs. And those bacteria can gain energy from light. Whereas chemotrophs are those bacteria which gain the energy from the chemical compounds. They cannot carry out photosynthesis. On the basis of electron source, it is again divided into designated as lithotrophs and organotrophs. Lithotrophs are some organisms can use reduced organic compounds as electron donors and are termed as lithotrophs. So those organisms which are using reduced organic compounds as electron donors are termed lithotrophs. That can be either chemolithotrophs or photolithotrophs. Next is about organotrophs. Some organisms can use carbon dioxide as electron donors and are termed as organotrophs. So the organisms use organic compounds. Some can be chemoorganotrophs and photoorganotrophs. Thus, the bacteria may be either photolithotrophs or organotrophs. So the photolithotrophs, these bacteria, they gain energy from the light, that is photo, and use the reduced inorganic compounds. They are capable of using the reduced inorganic compounds such as hydrogen sulfide as a source of electron. For example, Chromatium oceani. It is the organism in which those bacteria use reduced inorganic compounds as water. Next is photoorganotrophs. These bacteria gain energy from the light and use chemical compounds such as succinate as a source of electron. For example, rhodospirillum. So the photoorganotrophs means those gain energy from the light and use the organic compounds such as succinate. Next is about chemolithotrophs. These bacteria gain energy not from the organic compounds but from the reduced inorganic compounds such as ammonia as a source of electron. For example, nitrosomonas. Next is chemoorganotrophs. These bacteria gain energy from the organic compounds such as glucose and amino acids as a source of electrons. For example, Pseudomonas pseudoflora. Some bacteria can live either chemolithotrophs or as organotrophs. Like Pseudomonas pseudoflora, they can use either glucose or H2S and such as hydrogen sulfide as electron source. Next, on the basis of the carbon source, bacteria may be classified as I am showing. All the organisms, they require carbon in some form for use, for synthesizing cell components within the cell. Cell wall is essential. Some can use carbon dioxide as their major or even sole source of carbon. Such as organisms are termed as autotrophs. Other require organic compounds as their carbon source and are known as heterotrophs. Heterotrophic bacteria, for example. All the organisms require at least small amount of carbon dioxide in the heterotrophs. So coming to the autotrophic bacteria. This bacteria synthesize all their food from the inorganic substances. For example, water, carbon dioxide, H2S, salts. The autotrophic bacteria are of two types. Photoautotrophs. It means the bacteria capturing the energy of sunlight and transforming that sunlight into chemical energy. In this process, the photoautotrophs Carbon dioxide is reduced to carbohydrates and hydrogen donor is a water and produce free oxygen. Therefore, next is about photoautotrophs has chlorophyll pigment. So this photoautotrophs have chlorophyll pigment to perform the photosynthesis in the cell, and its main function is to capture sunlight. For example, for the photoautotrophs are cyanobacteria. Some photoautotrophic bacteria are anaerobes and have bacteriochlorophyll and bacteriovoidin pigments respectively. Next is about purple sulfur bacteria. This bacteria have the pigment bacteriochlorophyll and it is located within the intracytoplasmic membrane, that is thylakoids. This bacteria can obtain energy from the sulfur compound. So the name purple sulfur bacteria. Example, Chromatium, Theopedia, Rosae and Thiospirilla. Next is about green sulfur bacteria. These type of bacteria use hydrogen sulfide as a hydrogen donor. Reaction takes place in the presence of light and the pigment termed biovoidin or bacteriovoidin and bacteriophytin or chlorobium chlorophyll. So this bacteria utilize hydrogen sulfide and it also takes place in the presence of light. The next is this bacteria can take hydrogen from the inorganic sulfates. So they are capable of taking hydrogen such as sulfides and thiosulfates. Therefore the bacteria are known as photolithographs. Next is about chemoautotrophs. Chemoautotrophs in which the bacteria do not require any light. Similar to photoautotrophs, they doesn't require any light like that of the photoautotrophs. Here, chemoautotrophs require light, lack the light phase, but have the dark phase of the photosynthesis and the pigment for nutrition. So, this type of bacteria do not require light phase but have dark phase of photosynthesis. So the photosynthesis takes place in the dark phase and also the pigment of the nutrition. Many bacteria oxidize certain inorganic substances with the help of atmospheric oxygen. The reaction releases the energy which is used to drive the energy that is exothermic. So in the exothermic reaction, energy is released outside and that energy is used to drive the synthetic processes of the cell. Next is about sulfomonas. Those bacteria obtain energy by the oxidation of elemental sulfur, for example, or thiobacillus is an example for sulfomonas. Thiobacillus vegeatova, and this bacteria obtained energy by the oxidation of elemental sulfur. Next is elemental sulfur bacteria are the denitrifying sulfur bacteria oxidize elemental sulfur to sulfuric acid. For example, Thiobacillus denitrificans. Next is about sulfide oxidizing bacteria. This bacteria oxidizes the sulfur and releases the sulfur. Example, Vegeatova, that is 2 hydrogen sulfide plus 4 water is equal to 1 water plus 2 hydrogen sulfide plus 4 oxygen, which results in the formation of 2 water molecules plus 2 sulfur and some energy are released. So these are about sulfur oxidizing bacteria. Next is about hydromonas, that is hydrogen bacteria. This convert hydrogen to water. Example, Bacillus pandotrophus hydrogenomonous